In this video, we will discuss Vibrio, detailing its microbiological characteristics, its pathogenicity, as well as the main infections it causes and the corresponding preventive measures. Vibrios are gram-negative bacilli characterized by a typical curved, comma-shaped morphology. They are highly motile due to the presence of a single polar flagellum. Vibrios are common inhabitants of the human digestive tract and are also found in aquatic environments, particularly marine environments. These bacteria are responsible for cholera, a serious acute diarrheal infection. The species Vibrio cholerae, particularly serotypes 01 and 0139, is involved in epidemic forms of cholera due to its toxinogenic nature. The disease remains common in several Asian countries, while it no longer exists in Tunisia. Let's now talk about the classification of Vibrios. They belong to the Vibrionaceae family and the Vibrio genus. This genus includes several species of medical interest, mainly Vibrio cholerae, which causes cholera, Vibrio hemolyticus, and Vibrio alginolyticus, as well as other species associated with human infections, particularly those of digestive origin or related to the marine environment. The classification of Vibrio cholerae is based on the characterization of the somatic antigen O. Among the different serogroups, only 01 and 0139 are responsible for cholera epidemics due to their ability to produce toxins. Serogroup 01 is classified into two biotypes according to its biochemical and phenotypic properties: the classical biotype, which caused the first 6 pandemics since 1817, and the L-Tor biotype, responsible for the 7th pandemic that began in 1961. Serogroup 0139, identified in Bangladesh in 1992, is also an important epidemic strain. Let's now move on to bacteriological characteristics. Vibrio cultures are simple and quick to grow. They can be grown on ordinary media, as well as on blood-enriched media. Isolation is facilitated by the use of selective alkaline media, combined with adequate salinity, which promote the growth of Vibrios to the detriment of other bacteria. Furthermore, Vibrios are facultative aerobic and anaerobic bacteria. They have a fermentative metabolism and are characterized by positive oxidase activity, an important factor in their identification in the laboratory. The main substance produced, responsible for the pathogenicity of Vibrios, is an enterotoxin. It is a protein exotoxin, which plays a central role in the pathophysiology of cholera. It is thermolabile and has a characteristic structure composed of 5 B subunits, involved in cell attachment, and 1 A subunit, subdivided into A1 and A2, responsible for intracellular toxic activity. It is associated with extracellular enzymes that facilitate the penetration of bacteria into the intestinal mucosal lining. The environment is a potential reservoir for contamination and recontamination. Vibrios can survive for long periods in the natural environment, particularly after an epidemic, and can be found in sewers, fresh or salt water, and certain shellfish and crustaceans. Human contamination occurs mainly through oral ingestion of water and food contaminated by pathological excreta or poorly washed hands, which is the main mechanism of cholera transmission. The geographical distribution of cholera shows that this disease is not limited to a specific region. The first pandemic, which occurred in the early 19th century between 1816 and 1817, began in India and Asia before spreading throughout the world. Since then, there have been 6 successive pandemics, demonstrating cholera's ability to spread widely. Cholera is a notifiable disease. According to the World Health Organization, the global distribution of cholera shows a concentration of cases mainly in Africa and Asia, with a higher burden in resource-limited countries. This distribution reflects inequalities in access to safe drinking water and sanitation. For pathophysiology, the infection begins with the ingestion and attachment of the germ in the intestine. Contamination occurs by the oral route, mainly through the consumption of contaminated water or contaminated food, such as fruit, vegetables, and seafood, which serve as vectors for the bacteria. Pathophysiology of cholera involves 2 main steps. The first step is the attachment of the bacterium to the intestinal epithelium, mediated by pili. The second step involves the production and action of cholera toxin, which plays a central role in triggering the symptoms of the disease. The cholera toxin consists of subunits A and B with distinct functions. Subunit B is responsible for binding the toxin to specific receptors on the cell membrane, particularly GM1 gangliosides. Subunit A is subdivided into A1 and A2. Subunit A2 facilitates the passage of subunit A1 through the enterocyte membrane. Subunit A1, the active toxin, enters the cell and stimulates adenylate cyclase, leading to an increase in intracellular cyclic adenosine monophosphate concentration, which triggers the pathophysiological effects of cholera. An increase in cyclic adenosine monophosphate results in increased excretion of chloride ions, water, and electrolytes, sodium and potassium, into the intestinal lumen. Loss of water and electrolytes leads to severe dehydration of the extracellular compartment. We will now discuss the pathogenicity of cholera. Vibrio cholerae 1 is the causative agent of epidemic cholera. It causes acute intestinal toxin infection, strictly limited to humans. After an incubation period of 2 to 5 days, the disease is characterized by profuse diarrhea accompanied by vomiting and massive fluid loss, which can reach approximately 1 liter per hour, leading to a high risk of severe dehydration. The characteristic stools of cholera are watery and clear, sometimes containing mucosal debris, giving them their typical rice water appearance. Acute dehydration progresses very rapidly and can lead to death in 50 to 60% of cases if rapid rehydration is not provided. Now we will focus on the diagnosis of cholera. Clinical signs, combined with epidemiological data, often point to this disease. To confirm the infection, direct diagnosis involves several steps. First, samples are taken, either by stool culture, particularly from stools with the typical rice water appearance, or by rectal swabbing. Microscopic examination reveals comma-shaped bacilli with polar motility, while culture and biochemical identification confirm the presence of Vibrio cholerae. An agglutination test using a specific serum for Vibrio cholerae 1 confirms the serotype, and biotyping differentiates between the classical and L-Tor biovars, which is essential information for epidemiological surveillance and epidemic control. Let's now discuss preventive measures against cholera. Prevention is primarily based on hygiene measures, both individual and collective, particularly in countries with low socioeconomic status, to limit contamination through contaminated water and food. Initial treatment includes rapid rehydration and correction of acidosis, which are essential to prevent severe dehydration. Chemoprophylaxis may be considered in certain high-risk contexts, through the use of intestinal antiseptics or specific antibiotics. Finally, vaccination is a complementary tool, the injectable vaccine strain. Now let's talk about Aeromonas. It is a gram-negative bacillus of the Vibrionaceae family, mainly of aquatic origin. Regarding its habitat, it is mainly found in fresh or stagnant water, sea, food, and soil. This bacterium can be transmitted through contaminated water or food, via wounds, or by ingesting contaminated food. Infection with this bacterium mainly affects immunocompromised individuals. This bacterium can cause human infections, skin infections, diarrhea, septicemia, respiratory, urinary, peritoneal, or ocular infections. Thank you for your attention. We remain at your disposal for any further information or questions.